Hello everyone, welcome to the next episode of Micro Talks. Today we are going to be discussing the use of tissue culture for cultivation of viruses. Tissue culture is a laboratory technique where the cells or tissues are grown in a controlled environment outside of their natural biological context. It was first demonstrated by Enders, Weller and Robbins in 1949. They used poliovirus, which is actually a neurotropic virus, and they grew it in tissue culture using a tissue of non-neural origin. Let us see what are the different types of tissue cultures. You have organ culture, where small bits of organ they are taken by biopsy and they can be maintained in vitro for days and weeks. An organ culture is normally used for the isolation of some viruses which are highly specialized parasites. Example is the tracheal ring organ culture, which is used for the isolation of the coronavirus, which is a respiratory pathogen. Then you have the explant culture. In the explant culture, fragments of minced tissue, they are taken and they can be grown as explants. An example of the explant culture is the adenoid tissue explant. However, explant cultures are seldom used in virology. Cell culture is the next one, which is routinely used. Here, the tissues are dissociated into component cells and they are suspended in a growth medium. The tissues are dissociated by the action of proteolytic enzymes. Let us see how the cell culture is prepared. The desired tissue is removed surgically and it is mixed with a proteolytic enzyme like trypsin. Thereafter, it is shaken mechanically. The cells are washed, counted, and they are suspended in a growth medium. The growth medium essentially is composed of essential amino acids, vitamins, salts, glucose, a buffering system, generally a bicarbonate, and there is an atmosphere containing 5% carbon dioxide. This is supplemented with around 5% or calf or fetal serum. Antibiotics are added to this, obviously, to prevent bacterial contamination. Phenol red is used as an indicator of metabolic activity. Such growth media enable most cells to multiply with a division time of around 24 to 48 hours. The cell suspension can then be dispensed in bottles, test tubes or Petri plates. The cells will adhere to the glass and they will divide, forming a monolayer. What are the different types of cell cultures? You have three different types of cell cultures. You have the primary cell culture. These are normal cells that are freshly taken from the body and they are cultured. They are capable of limited growth and they cannot be maintained in serial culture. So example of a primary cell culture is the monkey kidney cell line, human embryonic kidney, and these are used for isolation of viruses for the production of vaccines. They can be passaged or subcultured only for a limited number of passages. Then you have the diploid cell strains. The diploid cell strains are cells of a single type that retain the original diploid chromosome number and the karyotype during serial subcultivation. They can undergo senescence after about 50 to 100 serial passages. Diploid strains are developed from human fibroblasts. This is an example, and they are susceptible to a wide range of human viruses, and very often they are used for the isolation of fastidious pathogens as well. Then you have the continuous cell lines. These are cells of a single type. Usually they are derived from cancer cells, and these are capable of continuous serial cultivation indefinitely. Good example of a continuous cell line would be the HeLa cell line. These are cells of the cervical cancer which have been isolated from the patient Henrietta Lacks. Then you have the Hep2 cell line and the KB cell lines. These cell lines can be maintained by serial subcultivation indefinitely, or they can even be stored in the cold till further use. Cell lines are also now used for the manufacture of vaccines. Let us now look at how we can detect the growth of viral growth in the cell culture. Detection of virus growth in the cell culture can be done using the cytopathic effect. Many viruses, they can cause morphological changes in the cultured cells in which they grow. And these changes can be observed under the microscope. These changes are called as the cytopathic effects, and the viruses that bring about these cytopathic effects are called as the cytopathogenic viruses. An example of cytopathic effect would be the crenation of the cells and degeneration of the entire cell sheet caused by enteroviruses, syncytial formation caused by the measles virus, degeneration foci caused by herpes virus, and large granular clumps that are formed which resemble a bunch of grapes caused by the adenovirus. Besides this, the viruses can also be detected using metabolic inhibition. In normal cell cultures, the medium will turn acidic due to the cellular metabolism. However, when viruses are grown in cell cultures, the cell metabolism gets inhibited and there is no acid production. So this can be identified by use of an indicator, for example, phenol red. So if there is metabolic activity, then the color of the medium will turn yellow, because phenol red is an indicator which has a color change from pink to yellow. If there is no activity, then the color will not change. The next criteria is heme adsorption. However, this is limited only for hemagglutinating viruses such as the influenza and the parainfluenza viruses, because they have the hemagglutinating spikes. The presence of viruses using heme adsorption can be indicated by adding 4% RBC suspension to the culture. If the viruses are multiplying in the culture, these RBCs will get adsorbed onto the surface of cells, and this is known as heme adsorption. Interference is another method of detection of viral growth. The growth of a non-cytopathogenic virus in the cell culture can be tested by subsequently challenging the same culture with a known cytopathogenic virus. So the cytopathogenic virus will bring about cytopathic effects. However, if no cytopathic effects are observed on challenging the same cell culture, which means the previously inoculated non-cytopathogenic virus has grown. Transformation is another method. This is limited again to the oncogenic viruses or the tumor-forming viruses. They induce cell transformation and loss of contact inhibition, so the growth will appear in a piled up manner and you will find the presence of microtumors. Immunofluorescence is a widely used method. Cells from virus-infected cultures can be strained by the fluorescent conjugated antibodies, and they can be examined under the UV microscope for the presence of the virus antigen. This gives a positive result, and therefore this finds a wide application in diagnostic virology. Thank you all for a patient listening. I do hope you enjoyed this episode of MicroTalks. Please like, share and subscribe to my channel so that you get updates of newer videos. Thank you so much.